My name is Joanna Romans, and I'm from Sick Kids Research Institute in Toronto. And my co-chair with me today is Samantha Alfonso from St. Michael's Hospital from the adult CF clinic there. And they have 450 adult patients at that clinic. So, okay, I'll try. Okay, I'll try to speak up. Is that better? Okay. Okay, so I'm Joanna Romans, and I'm from Sick Kids in Toronto. And my co-chair today is Samantha Alfonso, and she is from the St. Michael's Adult Clinic in Toronto, a clinic that has 450 adult patients. So in our session today, we have 4 speakers, and we'll introduce them as the presentations occur. But just before, I just want to give you a little bit of an introduction. First, to mention that the chairs have no disclosures. But I did want to remind you of just one fact, and that today the talk is on CFTR genetics. And I want to remind you that it's actually genetics that's 35 years old. CF genetics is older, but CFTR genetics is 35 years old. So it might be older than some of you in this audience and our co-chair. So when a discovery like when the discovery of the CF gene came out, like new discoveries, you know, sometime there's apprehension and anxiousness, as was first reported in a couple of the public newspapers in the United States, concerns with debates and anxiousness and in the public news. But also there was new hope, and that new hope related to the possibilities to help CF families. For the patients and to aid in the care of the patients, but also to provide effective and very good genetic counseling. And in fact, to go beyond helping CF families, but also to go to the population, because one could also think about carrier screening and other facets. So I did want to remind you about one thing with respect, though, to the discovery of the CFTR gene, and to remind you that it was a long time ago, and that in fact the discovery predated genome sequencing or all the amazing sequencing tools we have today. And that means that there are some legacy features, some of which you're familiar with the different nomenclatures that you have seen and perhaps been frustrated by. But nevertheless, that is both a privilege and a responsibility of genes in thinking about further care for a genetic disease. So CFTR genetics, of course, is largely based on sequence analysis, and certainly that pattern has worked in parallel technical achievement and sequence variant detection through the years. And now it is, of course, a very important tool in helping with the diagnosis and understanding of clinical presentations of CF and also CFTR-related disorders. So I just want to go back for 1 minute and have a reflection and thinking when you have a new gene, and the way the gene was identified was largely based on the study of families and using then molecular genetic analyses of the families. But one of the things you'd have to think about, because you want to understand what is the effect of the variant, what does that do? How does it define dysfunction leading to disease? You need to think about function. But when you define a gene based on genetics, you don't actually know that. But the one very earliest hint at function for the CF gene was in fact the expression pattern, and this is a very old gel now. Some of you may not recognize how to do it, but it did reveal that this method did show expression of this relatively large gene in tissues that were consistent with the disease. In fact, it was a part of the evidence that in fact this was the CF gene. The pattern made sense. You see expression in pancreas, nasal polyp, lung, colon, and in sweat gland, and a few other organs. And if we look today at gene expression, and all the single cell sequencing, you may be surprised to know that's still an evolving field, in the sense that I think the latest single cell paper I have seen now talks about 12 epithelial cells only in the proximal airway, 12 different ones. Some of them are related by development as defined by transcriptional patterns. And of surprise, a little bit of a surprise, some of them have no CFTR. Others do have CFTR, and of those that do, some of them have a lot of CFTR and some have very little. So all part of the ensuing effort to try to continue to understand CFTR. We also, at that time, very much in order to be able to interpret variants, had to have a way to do that. But as again, related to the way the gene was described, at best at that time, you need to think that there was no NCBI with databases of genes you could look up. That didn't exist yet then. In fact, there were only a few thousand genes really known, of which there were less than 1,000 genes, human genes known. So that was difficult to resolve. Although it was true, by looking at the very basic amino acid, it was possible to realize that the very first model in the papers, in fact, showed the transmembrane domains. You could predict that from the chemistry. There were also 2 nucleotide binding folds. That was evident by doing alignment, because even though there were very few genes known, a nucleotide binding fold was not a new feature in nature. And then there was that glob shown here with that big ball that's shown as a glob, and that's because that didn't align to anything. So if we look at this model, and now you look at those beautiful, eloquent models that you get from cryo-EM, which are just stunning and give us much more a dynamic sense of how the biogenesis of CFTR works and how, in fact, the protein domains fold and such. So it's actually not too bad a model. So these were important to set up and come up with function. At the same time as this work was being done, there was lots of data coming from electrophysiologists and cell biologists telling us, in fact, that we knew that if you looked at CF tissue and you looked at non-CF tissue, that there was dysfunction of fluid and ion flow. So those were important clues. Didn't exactly fit directly with this model, but at least it was a transmembrane protein, so somehow it fit in, and that's why the R word is there for regulator, because it wasn't obvious what it did, but somehow it had to be related to what the electrophysiologists were seeing and what the sweat test was telling us. So, but I did want to go back and think about variation again, and just to remind you that if you looked actually at the first paper, and the cover of Science had those many, many red horizontal bars, and that was to tell you, in fact, that was the delta F chromosome. In fact, we're looking there at chromosomal backgrounds of many people, all the people with the delta F chromosome, and that was the red one. But if you look further down, you actually also see those clusters of other colors. In fact, they were other chromosomal backgrounds, and the prediction at that time was, and a quote from the paper, and the data suggests that at least 7 additional mutations also contribute to the CF phenotype. But reality was that was off by orders of magnitude. And we'll hear today about how many causal variants now have been associated with causing CF, and we'll hear about some more as well. And just to press you forward on this, just to press you forward on this, and if you look at any gene today, and you were to look in the database and ask how many variants do we see if we look around the world, and this is a compendium of looking at CFTR compiled from multiple patient databases, and this is a project with Karen, who's about to speak in a few minutes. But looking at all the variation, there are about, just looking at the coding sequences in the exon-intron boundaries, there are in fact about 4,000 variants that you'll see. This is compiled from patient databases as well as population databases. And we're looking here at a very small piece of exon 20. This is only 18 nucleotides, and there are 22 variants. Okay, which is amazing. Not every nucleotide has a variant, but some nucleotides have more than one variant, particularly if you're a CPG, because that we know would have propensity to have a higher mutability than other sequences in the genome. But I just want to emphasize that if you thought there's a lot of variants now, it's not over yet. We still have a lot, okay? And we have to contend with this. But this brings us back now to our privilege and our responsibility of today, that we need to look at our learning objectives for a little bit, where we're going to hear from our speakers about CFTR2. We're going to talk about, I talked about, you have to understand what dysfunction means and what the variants mean, so we'll talk about CFTR dysfunction. Also going to talk about now the breadth and the broadening spectrum of what is CF and CF-related disease. And finally, we'll talk a little bit about the opportunity that we have to help families with CF and other related conditions. So we'll go now to our speakers. Okay. Oh, I'm doing the first speaker. So the first speaker is Karen Rarity from Johns Hopkins University, and she's going to tell us what's new with CFTR2.
Okay. Good afternoon, everyone. Thank you so much for the invitation to speak today. That was a really lovely overview of the last 35 5 years, if I do my math correctly, of CFTR genetics. And I'm thrilled to be here talking about the most recent update to CFTR2 and excited to share these new findings with you. These are my disclosures. So I am sure that most people in the room are at least somewhat familiar with CFTR2. I'm giving you a one-slide primer on sort of the background of the project and our goals. So this is an international initiative with the goal to analyze CFTR variants reported in patients from around the world, assign disease liability, and then make those interpretations and disease liability calls publicly available online. And you can find our website at cftr2.org. When you go there, this is what it looks like. You've got a search bar. I even think I have a pointer. Yeah. Search bar at the top. You can search for one variant. You can search for 2 variants. And then the website will tell you whether those variants cause CF, cause varying clinical consequences, which I'll define in a minute, or do not cause CF. These are our 3 categories. You'll note that the category of variant of uncertain significance is no longer a thing on the CFTR2 website. Those variants have all been recategorized. When we talk about the definitions of what we call variants, we think about everybody having 2 copies of CFTR. So when we think about how to define one variant, what we're thinking is that whatever's in the other gene is not contributing. So when we say in trans with a CF-causing variant, what we mean is that somebody has one allele with a null or very low function variant, and we're talking about the other one. So when in trans with a CF-causing variant, a CF-causing variant is expected to result in CF. Now we know that that severity can be variable, of course, but when in trans with a CF-causing variant, a variant of varying clinical consequences, or VVCC, results in CF in some people but not in others. Those people who don't have CF may be totally asymptomatic or subclinically not recognized, or may have a CFTR-related disorder or something in between like CRMS or CF-SPID. And then lastly, our category of non-CF causing is very specifically named non-CF causing, which differs from benign. A non-CF causing variant is not expected to result in CF, but a subset of people with some non-CF causing variants may have mild symptoms or evidence of a CFTR-related disorder. We do not expect those people to progress to CF if indeed all they have is a CF causing variant and a non-CF causing variant. I want to point out that when you go to the website, and I'm going to get into some nitty-gritty about how we do all this, but I want to just start off with reminding people of what is available to you and your patients online. When you go to the CFTR2 website and you search for a variant, you can get clinical information. You'll see here in this one that there are tabs along the top: summary information, sweat chloride, lung function, % with pancreatic insufficiency. This will give you clinical data from people with your variant of interest. And this is what patients and families are likely to see when they go to the website. But I want to point out something that I've come to know a lot of people don't know about. There's this little toggle line up here. It says, click here to switch to the healthcare and science view. So you can toggle between 2 different sides of the website. Now the website will default to what we call the general user view. That's what parents and families are most likely to see, and there's a lot of explanations about the data there. If you click over to the healthcare view, same clinical data available, but it's just displayed in a different way. So here you have one screen that shows you all the clinical information: sweat chloride, lung function, pancreatic status. Ooh. And you can also get information about functional testing and things like that. So 2 different sides of the website. Now you know how to toggle between them. And lastly, I'll just call your attention to some of the clickable options up in our search bar here. You can ask our team a question on a form. You can get a list of all the variants that have been on CFTR2 currently and in the past. You can find other resources. If you search for everybody's favorite variant, R117H or 5T, you can get information sheets on those as well. So lots of resources available to you on the CFTR2 website. Now, we're going to get into the nitty-gritty here of what CFTR2 has done over the years and the new updates. So throughout the years, we've had 3 different versions of CFTR2. The first one was released when the project piloted in 2012. This was 39,000, nearly 40,000 individuals collected from 25 countries and regions around the world. And with that data set, we were able to define the disease liability of 206 variants. Now, we are not at all creative with our naming, so I've called this one CFTR2 40K because that's how many people were in it. And that was released in 2012, of course, but the clinical data that was collected from registries went through about 2008. There's always a lag there to get the data into the registries, get to the registries to us, clean the data, et cetera. In 2015, we updated the patient data set to more than double what it was before. This is 89,000 individuals from around the world. We also upped the number of countries to 43, and with that data set, we were, over multiple years, able to finally interpret 804 CFTR variants and make those available online. And that data collection was released in 2015, but the actual data in it went through about 2013 at that time. I am thrilled to announce that earlier this week, the most recent update of CFTR2 went live with nearly 123,000 people from 55 countries and regions around the world. And we now have annotated and made available for search 1,167 CFTR variants. That data, of course, is being released in 2024, but the data collection, the clinical people that are in there, those data came through about 2019. Now, you'll note that some other things happened along these years. Oh, and I should note, throughout these processes of CFTR2, we've maintained the same methodology, which I'm going to dig into a little bit later. We look at clinical data, we look at functional data, and we look at population and penetrance evaluation. Now, as I said, other things have happened over the years since CFTR2 has existed, including most notably the approval and availability of CFTR modulators. One thing I do just want to point out is this most recent data collection goes through 2019. It is largely a pre-ETI or Trikafta or Kaftrio data set. Most of the individuals in the current CFTR2 data set that you are searching online, they may be on other modulators, but they're likely not on ETI. Now we're going to start out with what's in the new data set by way of individuals and worldwide representation. So one of the things we're really proud of is that this version of the database is markedly larger and more complete than previous data sets. In this one, we have about 70% of people reporting a sweat chloride value, 90% of people that report whether they are pancreatic sufficient or insufficient, 80% reporting whether they have Pseudomonas or not, 84% with an FEV1 measurement, 44% telling us whether or not they're on a modulator, and 16% indicating, yes, I am on a modulator or I have been on a modulator. From a genotype perspective, the data set is getting more complete and better annotated from a registry perspective over the years. If we look at just the active patients, so those that are living in the most recent years of data collection, 92% of them have 2 variants recorded. Only 3% have neither variant reported in the data set. Here is a world map of all of the countries or regions that have contributed to CFTR2 in this current data set. These are colored by just the size of the contribution, so not prevalence in that country, but just the actual sheer data size. This is a very busy slide, but I've put all the numbers up there so you can see the darker countries like the US, that's a contribution of over 50,000 patients. The ECFS contribution is also quite large. It's a little under 39,000 patients. That is an amalgamation of about 23 different countries. But you can see that we have representation from Asia, not a lot, but we do have representation from Asia, we have representation from Africa, we have representation from South America, North America. So if we come back to this sort of grounding slide here, I want to talk about the CFTR2 methodology because this has really remained unchanged over the years, though it has evolved a little bit. The first thing we do when we want to figure out how to interpret variants is we got to populate the data set. So I already told you we did that. We got 123,000 people. So now we go into that data and we look at all of the genotypes that are reported and we just make a list of the variants and put them in decreasing frequency order. Delta F508 is way at the top, and then variants reported in one person are at the bottom. Then we analyze the variants. I'm going to explain this in more detail coming up, but we look at 3 main pieces of data: a clinical evaluation, functional evaluation, and population or penetrance evaluation. Hopefully, those 3 pieces of data agree, and we can then annotate the variants online as I described earlier. So as Joanna mentioned, we have 1,085 variants that we have interpreted to cause CF, 55 that cause CF in some people but not others, and 27 that we have evidence showing do not cause CF. So how do we do these three pieces of data evaluation? So when we talk about a clinical evaluation, the number one thing we need to do is identify patients that have usable clinical data. If we have a particular variant of interest, again, we want to look at that variant on a background of something that's really not contributing to the phenotype. So we look for patients who have our variant of interest, in this example, E474K, and then something else that we know to have very low function. Conveniently, lots of people have the F508del variant, and also conveniently, for our purposes, it does not contribute much function at all. So we usually have a good number of people that we're able to look at. We evaluate the mean sweat chloride of this group of people with our variant of interest. If we don't have enough sweat chlorides to look at, we can evaluate whether these individuals are reported to be pancreatic insufficient or not. Do they have clinical evidence of CF? When we come to the functional evaluation, this is much more complicated. I'm going to try to distill it, or at least maybe tell you about the complexity, but not delve too far into it so that I don't get yelled at for going over time. When we think about a functional evaluation, we want to know what a variant does. And it seems very simple to just say, you got a variant, just determine the effect. Like, how hard can it be? Does it do something to CFTR? And that's like a really nice way to think about it. But in reality, we have to ask a couple questions. Is the variant intronic and does it affect splicing, or is it exonic and does it affect translation? If it affects splicing, is there any normal splicing or no normal splicing? If it affects translation, is there any protein synthesized or no protein synthesized? Then we can look at whether there's stable protein that results from splicing. Then we can look at whether protein that is produced is improperly trafficked, partially trafficked, or properly trafficked. And if so, how does that function look? So some of these are RNA effects, some of these are protein effects, and we have ways to address different parts of them. What I'm trying to convey to you here is that we have a basic framework for how we interpret different types of variants, or how we do functional assessment on different types of variants. So we can sort of funnel things into the right direction once we can answer these questions. And there are some variants that require multiple types of functional evaluation because they may have multiple effects. Lastly, we come to our population or penetrance evaluation. The first thing we do, if we can, is evaluate whether there is any evidence of non-penetrance for a variant, meaning, does this variant occur in somebody who also has a CF-causing variant and who does not have any clinical disease? We did this originally with CFTR2 by looking at fathers of individuals with CF who had one CF-causing variant that they passed along to their child and who also had something else. And if those fathers did not have any evidence of infertility or clinical disease, that was good evidence of non-penetrance. Now, we can't repeat that study because we've gotten into so many very rare variants. We'd have to have a population of hundreds of thousands of fathers, and we just don't. But we do have population data sets that can give us information about the frequency of a particular variant in the general non-CF population. And that can give us information about how likely that variant is to cause CF. We use the NOMAD data set. We've also, in the last few years, developed a very fancy math algorithm that I can partially explain, that uses a Bayesian framework to look at frequency of a variant in CFTR2 versus frequency of a variant in NOMAD, and then make a call on the likelihood that that variant will cause CF. And so that actually gives you a percentage score. Low scores are unlikely to cause CF, high scores are likely to cause CF. I think examples are most helpful, and so I want to show you the type of data that I actually show the CFTR2 team on a regular basis and say, how are we going to do this variant? So this is a frame shift example. This is a legacy name, 3617del-GA. First thing we do, go into the data set and find people who have our variant of interest in trans, or presumably in trans, with a variant not contributing to the phenotype. Let me be very clear, real variant, fake patient data. Now in this fake patient data, we have 2 people with very diagnostic sweat chlorides and 2 people reporting pancreatic insufficiency. This is all very consistent with a CF-causing variant. Frame shift variants don't, at this time, require functional evaluation for us to determine that they are very likely to result in no or very low function, so we don't actually functionally test these. We do on occasion if there's good reason to, and there are exceptions to that rule. As far as I can tell, this is not one of them. So we assume that this is going to lead to no CFTR protein that's consistent with a CF-causing variant. Lastly, we look at allele frequency between CFTR2 and NOMAD. We get our Bay-PR score. This one is quite high. That is also consistent with a CF-causing variant. This one is relatively straightforward because those 3 pieces of data all agree with each other. This one gets the rubber stamp of CF-causing. I'm going to show you one other example that's a little bit less straightforward. This is a missense variant called Q237E. Here's the clinical data, fake patient data. We have our people with Q237E and another variant that we think is not really contributing. Now, we do have a mean sweat chloride of this group of 66, which is above the diagnostic threshold, though if you look at those values, which maybe you can do, you can see that there's a broad range. The lowest one is 44, highest one is 80. So we are actually straddling that diagnostic threshold with the range of data here. And everybody that reports their pancreatic insufficiency status says, nope, I'm pancreatic sufficient. This is consistent with a CF-causing variant, but it's definitely a little mild or has residual function. And indeed, when we do the functional evaluation, that's what we find. This particular variant was tested both in CFBE cells and in FRT cells and has similar function in both. 12.5% chloride conductance, that's on a 0 to 100% scale of percent of wild type, and 16% chloride conductance in FRT cells. Our functional threshold for something that causes CF is typically below 10%, so this is above that, but not by much. These values are most consistent with a variant of varying clinical consequence. When we look at the allele frequencies, this one is actually, I don't want to say common in NOMAD, but it's got some alleles there. So we've got 6 count of this variant in CFTR2, 20 count in NOMAD. This actually gives a BAPR score that is quite low and consistent with a non-CF causing variant. So here's where we have 3 pieces of data that seem to maybe contradict each other. However, when you look at the nitty-gritty, what this is really telling us is that this is probably just a borderline variant. We've got sweat chlorides that are not so far over the diagnostic threshold, and some of them are actually in the gray zone. We've got chloride conductance. At least one of these values has overlap of that 10% threshold when we look at the error. And we've got evidence that maybe not everybody in the world with this variant would have CF. And so this gets the stamp of variant of varying clinical consequences. All right, now I'm going to talk about the number of variants interpreted and where we're going with that. So, as I said, we've interpreted over 1,100 variants now online. When we started, or even pre-CFTR2, if you just interpreted one specific variant called Delta F508, you could account for about 70% of alleles. As you up the number of variants you interpret, you can account for more and more alleles. So going to the ACMG23, the original CFTR2 159, 485, 804, and now 1167, what you can see is that 1,167 variants accounts for about 98% of alleles that have been reported to us. But you can also see that it's taking a lot more variants to move the needle and close that gap. So 86% of variants remaining for interpretation are reported in less than 5 people. They are large in number, but not huge when it comes to allele frequency. So I'm going to kind of finish up here with where are we and where are we going in the future. So in this newest data set, we have 2,891 unique CFTR variants reported. And if I do my math right, that is more than 7, as we learned about. So there's more than 7. Now, of that 2,900 reported to us, just under 2,100 are eligible for interpretation. I'm going to come back to that in a minute. We have about 800 variants reported in the data set that we are not able to interpret because we cannot decipher the nomenclature. I'm just going to make a quick note of kind of what this looks like. These are variants that very likely somebody tried to enter something and there's just a typo in it. That accounts for about 1,500 people. Again, 1,500 is not a small number, but it's about 1% of the database. So not a huge number of people, but I'll just put a note out for anybody in the room who works for a registry. If you'd like help interpreting or deciphering some of these variants, we would be very happy to try to point out and say, oh, this one, we don't know what this means, or this is confusing to us, and things like that. So we're going to put those 800 variants aside and focus on the 2,100 or so that we need to interpret. I just told you 1,167 are online. That means we have 926 left. Now, what's a little bit challenging is when I think about the data that we have available to us, we're looking for clinical data, functional data, and population or penetrance data. There's about 200 variants that have either none of those available or just one. These are really challenging. So it may be that we are going to require additional information from registries, a new clinical data collection, different type of functional studies. We may not be able to do much with this group as it stands right now. What we do have, though, is over 700 variants in our remains for interpretation group where we've got 2 or 3 pieces of data. And I can tell you that for variants in the 2 group, there's functional testing underway at the CFF Therapeutic Lab that's going to move those 2 variants into the 3 category. And then in an ideal world, if we combine those 700 or so variants with the ones we have online, it is very likely that we will be able to use the new data set to interpret close to about 1,900 variants. Now, there's going to be complicated ones where those 3 pieces of data don't align and we need to do additional studies to reconcile them, but I do believe we're going to be able to at some point. I know, I'm running. I know. So I want to just give an analogy here because I've mentioned that the methodology has really remained the same even as principles and tools have evolved. So I sort of think of the original CFTR2 data set as like an old Apple computer. So it got the job done. It wasn't super fast, but it was kind of clunky and we were learning as we went along. Then we got to the 89,000 person data set and it was more like a laptop. It was a little smoother. There were improvements made along the way, but I think now we're in the smartphone era here with this 123,000 person data set. And there are many people in this room probably that have contributed to these changes along the way. So even in the initial CFTR2, we started using expression mini genes to evaluate splicing. That was a new tool that we had for functional evaluation. In the second version, we had registry QI initiatives that improved the quality of the data collection. That really helped us with the clinical evaluation. NOMAD was founded. We used to use a population data set called 1,000 genomes. It was literally 1,000 genomes, that's it. And in this new data set, we have further improvements in database systems and data capture for the clinical side of things. NOMAD version 4 was released. So I said we used to use this population data set of 1,000 people. NOMAD 4 has 800,000 people in it. So we have a much broader population that we can use for frequency evaluation. We now have the availability of organoids and primary cell studies when we need them. And we have, thanks to the CFF Therapeutic Lab, high throughput variant testing to help us with the functional evaluation. So all of these things together have really brought us into the smartphone era. And I have to say, I think if you had asked our team when we started, do you think you could get to almost 2,000 variants online? We probably would have laughed and said, I just cannot imagine that we would interpret rare variants like this. We're down to variants that have been seen in 1, 2, 3 people worldwide. I just don't know that we would have thought it possible, maybe even 5 or 7 years ago, but with the development of these new tools and really maintaining our same robust principles, we're headed, this is my analogy here, we're headed toward the smartwatch era here, being able to do this. I will end here and say just a huge thank you to the CFTR2 team who is nice enough to put up with me every Friday at 9 a.m. for over a decade. This is just a really wonderful group of people. Joanna's up there. This is a really wonderful group of people that think very hard about CFTR. You could not find a harder working, nicer group of individuals with more expertise in CF and CFTR. It's really incredible and I'm privileged to work with all of them. Many, many, many other people to thank. I'm not going to be able to read all names. The whole team at Johns Hopkins, the CFF Missense group, certainly collaborators at many labs around the country and the world, the CFFT, UT Southwestern, Rosalind Franklin, Emory Vertex, University of Bristol, and perhaps overarching all of this, all of the CFTR2 registry and clinical contributors and patients and families, without which this project would just be a whole big nothing. It just wouldn't exist. So thank you very much. I may or may not take questions.
Thank you, Karen. So we have 2 minutes for questions if anybody has anything. Otherwise, we will have a panel of everyone, all of the speakers at the very end. So if anybody wants to submit their questions online, we will have time to answer those at the end as well. Yeah, we do have 2 minutes if anybody has anything right now. No? Okay, wonderful.
Fantastic.
Thank you.
Thank you.
Okay, so I'd like to invite Dr. Beeler. Dr. Beeler is a research fellow at the Cystic Fibrosis Foundation Therapeutics Lab in Lexington outside of Boston, and he will be discussing today functional serotyping and drug testing.
Well, yes, thank you for the introduction and for inviting me here today to speak about functional serotyping and drug testing of rare CFTR variants for effective modulator intervention. I have nothing to disclose, so there are 3 parts to this presentation, and the first will address the challenge of rare CFTR variants and the rationale for in vitro functional serotyping of rare variants. In the second part, I'm going to present data from a study that we published earlier in the Journal of Cystic Fibrosis that investigated the responsiveness of 655 CFTR variants to elexacaftor, tezacaftor, ivacaftor, which are the components of Trikafta. And in the third part, the focus will be on interpretation of the data from the study and implications for people with cystic fibrosis who might benefit from but are currently not approved for modulator therapy. I assume many are familiar with the term theratyping. Just to be clear, in the context of this presentation, theratyping means in vitro testing of CFTR variants for their responsiveness to CFTR modulators for the purpose of identifying CF-causing variants that could be treated with modulator therapy. So there are over 2,000 variants of the CFTR gene, and many of those have only been observed in a few people with cystic fibrosis, as you can see here by the histogram of allele counts from the CFTR2 database, which also includes the allele frequencies of all 655 CFTR variants that we have studied. And this makes drug approval for rare variants based on traditional clinical trials impossible. So fortunately, in 2017, the FDA started to allow in vitro data to be used for label expansion to include rare CFTR variants. And this was possible because Vertex had already established the safety of the drugs in clinical usage and convinced the FDA that their in vitro assay using fish or rat thyroid cells, short FRT cells, stably expressing CFTR variants is a validated and robust system and predictive of a clinical benefit. Now, building on the Vertex experience with the FDA, the CF Foundation decided to sponsor a large-scale serotyping study that was conducted at the labs of Dr. Bob Bridges, Dr. Phil Thomas, and the CFFT lab headed by Dr. Martin Menze. And each lab screened 655 CFTR variants for their responsiveness to elexacaftor, tezacaftor, ivacaftor in transiently transfected FRT cells. And now we chose this approach because transient transfection of FRT cells with CFTR cDNA is a flexible and scalable method to serotyping of a large number of CFTR variants. Now, almost 4 years ago, the FDA expanded its approval of 3 CFTR modulator therapies to include 183 CFTR variants, enabling access to highly effective modulator therapy for most people with cystic fibrosis in the United States. But there are about 6% of people with cystic fibrosis in the US, which are represented here by their gray slice, that currently do not have available therapies for the underlying cause of their disease. And many of those have variants that are not amenable to modulator treatment. But we also know some of them harbor variants that can be treated with modulators. So our goal is to identify additional CF-causing variants that are not FDA approved, that can be treated with modulators using in vitro assays like the FRT cell system to further reduce the size of that gray slice as much as possible. Now, unfortunately, our FRT cell system as currently designed does not allow serotyping of intronic, splice, promoter, and nonsense variants. So Gary Cutting and Karen Rare, they put together a list of over 650 exonic CFTR variants that are expected to make CFTR protein. They can potentially be targeted by modulators. And the majority of those, as you can see here, are missense mutations, but we also included some in-frame deletions and complex variants comprising 2 or more gene mutations on one allele. And at the bottom of this slide, you can also see the location of the studied variants along the primary sequence of the CFTR protein, which also highlights that the selected variants occur throughout the full coding sequence of the gene. I want to highlight poster 337 here. Now, the poster summarizes a preliminary analysis of 272 CFTR variants with the goal to evaluate their responsiveness to the new Venzacaftor triple. So in the next few slides, I intend to briefly go over the experimental design of our study and then share some key findings. And what you can see here is the plate layout of a 24-well assay plate. Each well contains FRT cells that express CFTR protein, whose chloride transport function can be measured by an electrophysiological setup that allows recording of multiple plates in parallel. And each assay plate also contains intraplate controls for wild type G551D and F508del, as well as 4 blinded variants. Now, 24 hours before the assay, elexacaftor, tezacaftor was added to the wells that are shown here in the green background, and matching concentrations of DMSO were added to all the other wells. All the treatments were done in duplicates at each of the 3 sites, giving a total N of 6 for every blinded variant tested. Now, what you see on the left are representative conductance traces for wild type G551D and F508del CFTR that were obtained from one of the corresponding wells, circled here in red. And forskolin was used to stimulate CFTR, with wild type CFTR shown in black, giving the largest response, followed by F508del treated with elexacaftor, tezacaftor. There was no forskolin response for untreated F508del and G551D. And the red trace also shows that the gating defect of G551D can be rescued by the addition of ivacaftor, which also further potentiates wild type and rescued F508del activity. And we also added an inhibitor cocktail to ensure that the signal was specific to CFTR. What you see here on the right are representative conductance traces from a modulator responsive variant that was treated for 24 hours with either DMSO or Alexa tezacaftor. And the assay was designed to give individual and combined readouts for Alexa tezacaftor and ivacaftor. But for the purpose of this talk, the important readouts are highlighted here by the thicker arrows. And so this black arrow here represents the forskolin response in the absence of modulators, which is often referred to as baseline activity or baseline function, and indicative of disease liability. And the thick red arrow here shows the gain of function from baseline in response to Alexa, Tisa, ivacaftor, which is a measure of a variant's modulator responsiveness. And yes, we averaged all the individual responses and normalized them to the in-plate wild-type forskolin response, and all those data can be found in the supplement of our paper, together with protein expression data from Western blots. Okay, so let's have a look at some of the results. What is plotted here on a logarithmic scale is the baseline function for all 655 CFTR variants as % of wild type. And every variant baseline function is represented here by a black dot, and 342 of those you can see are within that gray area, that gray box, meaning that those have less than 10% of wild type function. And a combination of variant alleles with low baseline function, is generally associated with a more severe disease outcome. Now, we also identified 104 variants with more than 50% of wild-type baseline function. Those are likely not CF causing, and the majority of those have an allele count of 1 in the CFTR2 database, and it is possible that the actual pathogenic variant was missed at the time of diagnosis. There's also a few variants in our collection whose pathogenicity might not be fully captured by our cDNA model, as they may affect splicing, and we flagged those in the paper. Okay, so this plot shows the responsiveness of 655 CFTR variants, indicated here by the red triangles. And each red triangle represents a variance improvement in function from baseline as % wild type. And there are 543 of those that respond to Alexa, Tisa, ivacaftor by an increase of 10% of wild type function or more. And among those are 231 with less than 10% of wild type baseline function. And those are represented here by the red triangles and the green box. Now, Vertex and the FDA agreed that an in vitro improvement of 10% of wild-type function likely confers a clinical benefit at the population level. But we also know that people with cystic fibrosis with very low baseline function might actually experience a meaningful benefit, a meaningful clinical benefit if their baseline function only improves by a few percent. So let's have a look at the variants that are not approved for modulated treatment. There are 376 of those that responded to Alexa Teser-Ivacaftor by an increase of 10% of wild-type function or more, and 152 of those have less than 10% of wild-type baseline function. And again, those are represented here by the red triangles in the green box. We also identified an additional 140 responsive variants, and this brings us to a total of 292 responsive variants with less than 50% baseline function that are not approved. And I think this is an impressive result and encouraging for people with cystic fibrosis who might be able to benefit from those modulators. Okay, let's also have a brief look at the 174 Trikafta approved variants that we tested in our study. And we find 97% agreement with Vertex or the FDA label. There's only 5 variants that clearly miss the 10% responsiveness threshold, and those are the red triangle circled in white here. And I think this high level of alignment is impressive, considering that we used a different assay and the data were generated in 3 different labs. It also provides confidence in the data, and it suggests that the transient transfection method is of utility in rapidly assessing new variants as they are discovered. Okay, so we also thought of another way to break down variants taking into account measurement error, rather than dividing them into responsive and non-responsive variants based on their mean response values. And to do that, we used an empirical base method and calculated the 90% credible intervals, which are shown here as vertical lines for every mean response to variance mean response to Alexa Teser-Ivacaftor. And this allows us to divide those variants into 3 categories. Responsive, highlighted here in blue, whose lower bound of the 90% credible interval is above the 10% mark. Non-responsive variants highlighted here in red, whose upper bound of the credible interval is below the 10% mark, and borderline responsive variants, whose true response could be less or more than 10% of wild type. And I want to point out that the majority of the borderline responsive variants has a mean response value that's far above the 10 percentage points of wild type CFTR function, and many of those are FDA approved, as indicated here by the orange lines at the bottom of this graph. Now, anyone who is interested in the variant ideas associated with those colors can find those in table 2 in our publication. So what have we learned? I think the data from this study should further strengthen our faith in cDNA-based in vitro assays as a valuable means for teratyping of CFTR variants. Our approach, however, is a mutation-specific approach that differs from but complements personalized teratyping efforts, which use cells from individuals to predict their own personal clinical benefit. Therefore, careful consideration should be taken with the interpretation of the data from this study. For example, our cDNA-based model does not fully represent the processing of the native CFTR gene, and therefore, data that are associated with variants that may affect splicing must be interpreted with caution. And second, cDNA-based models are more likely to predict a population-level response than predictive for the response in an individual that is homozygous for a given variant. Also, evidence based on real-life data suggests that the threshold of 10% functional improvement relative to wild-type function should not be considered as a rigid cutoff for modulator access. For example, McCaig and others have shown that people with cystic fibrosis with genotypes that confer less than 2% of normal function sees their lung function measurement decline to less than 60% predicted FEV1 around 27 years of age. That's 35 years earlier than people with cystic fibrosis with genotypes that confer 5 to 10% of normal function. Another example, N1303K missed the proof of responsiveness threshold in our assay, and yet there are multiple clinical studies from several countries that report that people with cystic fibrosis harboring N1303K variant experience significant benefit from Trikafta treatment. We have shared our data with the drug sponsor, Vertex Pharmaceuticals, and Vertex is using this information to inform their own internal research into whether to submit an application to the FDA for modulator approval of new variants. But what are the options today for people with cystic fibrosis with responsive variants that are not yet approved for modulator therapy? Well, we encourage people with cystic fibrosis who carry responsive variants to discuss off-label access to treatment with their care team and potentially with their health insurance providers. And people with CF may also be eligible to have personalized serotyping performed in support of drug access. And there are sites and programs that are committed to helping people with cystic fibrosis who could benefit but don't currently qualify for a modulator to gain access to highly effective modulator therapy. And I'm just highlighting a few examples here. Okay, so in summary, our goal is to enable serotyping, to use serotyping to enable access to modulators for all people with cystic fibrosis that could potentially benefit from modulator treatment. And to that end, we characterized 655 variants for their responsiveness to Alexa, Tisa, ivacaftor, and identified 292 variants that are not yet approved with less than 50% baseline function. And looking ahead, I am hopeful that more people will benefit from the arrival of new modulators, as well as from approval of additional variants. And as I said, we currently conduct the second round of serotyping at the lab, and based on preliminary analysis, nearly 90% of variants, of over 200 variants that we studied to date, are responsive to the vanzacaftor triple, which currently undergoes priority review for FDA approval. And to give you one example, so we tested vanzacaftor ivacaftor in cells from an individual homozygous for W1282X, which is a nonsense variant that makes a truncated version of the CFTR protein that is poorly functional but can be rescued by modulators, as demonstrated here in this graph showing functional rescue of W1282X by vanzacaftor ivacaftor and Trikafta. And you can see that vanzacaftor ivacaftor is about threefold more efficacious than Alexa-Tisa ivacaftor in inducing a swelling response in intestinal organoids in this assay. We also plan to test those variants I just talked about today that did not make the cutoff for responsiveness with Alexa-Tisa ivacaftor to see if those are treatable with the vanza triple. Okay, and I think this is all good news for people with cystic fibrosis. But we also know that new treatments cannot come soon enough for those who currently don't benefit from modulators. And to address that unmet need, the foundation is investigating and leading research to advance genetic therapies to provide transformative treatments to people with cystic fibrosis who are ineligible for modulator treatment, and eventually to everyone with cystic fibrosis. So, and this is my last slide. This truly was a team effort, and I want to thank everyone who contributed and supported this study. And thank you for your attention.
We do have 5 minutes for questions. If anybody has a question, please come up to the microphone in the middle. The session is being recorded and is being translated into Spanish. So if you have a question, please come and speak in the microphone.
Hi,
Hello?
my name is Cambry White, and I have a son with, he has cystic fibrosis. He has 2 rare cystic fibrosis variants, and they are not eligible for Trikafta. However, we do have access to Trikafta because of off-label access, so that's great. I have a comment and a question. My comment is that I really appreciate you highlighting the fact that there are CF variants that can potentially provide clinical benefit, even if the response is under 10%. My question is, how did you guys agree on the 10% threshold to label one category responsive and the other one non-responsive?
So we did not. That was an agreement between Vortex and the FDA.
Okay.
Yeah. With we, I mean the Cystic Fibrosis Foundation Lab. Yeah, no.
Okay, thank you.
So I have a question. So in terms of moving forward, when's the next set arriving? We have 272 more, but
It's a question for Karen, I guess. Well, we test
oh, you're
everything.
waiting for Karen. Okay.
Yeah, we
we'll work
test
on that.
everything we get our hands on. Yeah. Yeah, I mean, just bring it our way. We'll screen it. Yeah. We're emotional right now. Yeah, so. Yeah.
Okay.
All right.
Thank you. Up next, we have Dr. Elizabeth Tullis. Dr. Tullis is our medical director at the Toronto CF clinic, which is the largest adult CF clinic in North America. And so I'd like to welcome Dr. Tullis.
Thank you. So thank you very much to the organizers for asking me to speak today. I'm going to be talking about CFTR-related disorder and addressing sort of the classification and definition, and then talking a little bit about challenges and the knowledge gaps in this disorder. I have no relationships to disclose that are related to this presentation. So I don't need to tell this audience that CF is a multi-organ, autosomal recessive, one single gene disorder that is a result of defective function of the CFTR protein causing clinical phenotype. And I think also that this audience is familiar with this idea of a spectrum of conditions that are related to CFTR function or dysfunction, with cystic fibrosis being at the lower end of the scale, with CFTR function below 10% wild type. And within the group of people with CF, there is also a spectrum with those with the lowest amount of CFTR function having more severe disease, as defined by pancreatic insufficiency, and as a population, more severe lung disease at a younger age. Clinicians also have identified for decades now that there's conditions where there's the clinical features of CF, and these people also have an increased prevalence of CFTR gene variants. And the clinical situations that come up like this are male infertility with congenital bilateral absence of the vas deferens, recurrent acute pancreatitis or chronic pancreatitis, and bronchiectasis. And in the past, there was no standardized terminology, so it was all very confusing. People would talk about patients having atypical CF or non-classic CF or pre-CF. And so there was a variety of different terms, and everybody was meaning something different. Some people were meaning CF, and atypical CF was pancreatic sufficient, and other people were referring to this other situation. And then in 2011, there was a European consensus conference that was then updated in 2022. And at that initial consensus meeting, the term CFTR-related disorder was coined to describe a clinical entity that was associated with CFTR dysfunction, but did not fulfill the diagnostic criteria for CF. So the next question is, okay, well, what does that mean and where does this fit on the spectrum? So we're looking at CFTR-related disorders as falling with approximately levels of 10 to 30% of CFTR function, a bit of a gray zone, obviously, but fitting in with more function than people with CF, but less function than obligate heterozygotes. Now, we're also all familiar with the diagnostic criteria for CF, where you have a clinical phenotype, and then you also have to have diagnostic tests that show either evidence of CFTR dysfunction, and the level of dysfunction that is shown on the sweat test to diagnose CF is a sweat chloride of greater than 60 millimoles per liter. And then either or evidence of 2 CF-causing gene variants in genetic testing. When we look to the diagnostic criteria for CFTR-related disorders, we have a very similar sort of template. We have a clinical phenotype that has expanded from the original 3 kind of common conditions of CBAVD, bronchiectasis, and pancreatitis to add other conditions that have been noted to be similar to those seen in people with CF, but where there is an increased frequency of CF gene mutations. Now, when we look at the diagnostic tests, there has to be evidence of CFTR dysfunction. But whereas when we're looking at CF and using the sweat test to determine the difference between people with CF and people who have no gene mutations, the sweat test is very good at doing that. But now we're looking at a more nuanced measurement of CFTR function. And the sweat test itself is not adequate on its own because in these gray zones, it's not so clear at distinguishing. So there are additional functional tests for CFTR that include intestinal current measurements, which is an ex vivo test that uses intestinal cells from a rectal biopsy that are studied to look at chloride conduction. The other test of CFTR dysfunction is the nasal potential difference test, which is in vivo, so it's done directly, examining the conductance of chloride and other ions across the nasal epithelium in a person that's being studied. Now, when I gave you the diagnostic criteria for CFTR-related disorder, I didn't say anything about the genetics. However, in this case, CF genotype is sort of a surrogate marker of CFTR function. So, when we look at this template and we say, well, what does that translate to in terms of when I'm trying to make a diagnosis? We've got the clinical phenotype. And then when we have evidence of CFTR dysfunction, that would be a sweat test that is above the normal range of 30, but below the CF range of 60, so between 30 and 60. But because the sweat test in and of itself is not sensitive or accurate enough to distinguish in all of the patients, the diagnosis of CFTR-related disorder requires evidence of dysfunction in at least 2 different CFTR tests. Now, if we're looking at the genetic side of things, one option for the diagnosis of CFTR-related disorder is 2 CFTR variants that are known to reduce CFTR function. So these would be your variants of varying clinical consequence. And you can either have 2 of them, or you can have 1 and 1 CF-causing variant. Obviously, if you have 2 CF-causing variants, that's diagnostic for CF. And then when you're in a situation where you can only find 1 variant that's known to reduce CFTR function, then you need to have the functional tests. So you can see it's a little bit more complicated than the diagnosis of CF, and we'll talk in a few minutes about some of the challenges. In the updated CFTR-related disorders sort of guidelines, they have a nice algorithm that starts with you need symptoms to start, Then you do genetic analysis and a sweat test. That allows you to identify some people with a borderline sweat test. Remember, we need 2 levels of CFTR function. So then we do a second test, which is either NPD or intestinal current measurements. And if they're in the range of, that would be the amount of dysfunction that would be compatible with CFTR-related disorder, then you have a diagnosis. If in your initial genetic testing you have 2 variants of varying clinical consequence, or 1 CF causing and 1 VVCC, then you can make the diagnosis of CFTR-related disorder. And even though this term is no longer in CFTR2, if there is one variant that's either considered not CF causing, then you have to move over and look at your functional tests. So this is how the algorithm goes. So then, knowing the classification of this disorder and how the diagnosis is made, I'm going to move on now to talk about some of the challenges and some of the knowledge gaps that are related to this condition. The first one's pretty obvious, and these are challenges related to the diagnostic tests. So sweat testing is pretty generally obtainable in CF centers, but nasal potential difference and intestinal current measurements are only available in specialized CF centers. And so here, for example, in the publication of the guidelines, they mentioned that MPD is only available in about 30 centers in North America, in 15 in Europe or Israel. So this is not broadly available. And although there are standardized operating procedures for MPD and in ICM have been developed, there isn't yet an international consensus on interpretation of what the thresholds would be in these tests to call something CFTR-related disorder. So different centers have developed their own threshold measures, and there are some ones that have been created and validated, but there isn't yet an international consensus. And so this leads to some challenges in our ability to discriminate between CFTR-related disorder and carriers, or CFTR-related disorder and more severe dysfunction, or CF. In addition, there are other measures of CFTR function, like the beta-adrenergic sweat test, which is not yet, is part of the consensus, been agreed to as one of the possible tests to measure CFTR dysfunction. The second issue is, well, you could say, okay, I don't have access to these additional tests of CFTR function, but what about using genotype as a way of making a diagnosis? And that, of course, is very possible, but you would need to do full sequencing. And so you couldn't rely on just a panel of the most common mutations. And so some centers don't have access to full sequencing in some countries in order to do this. So there are challenges with making the diagnosis. Now, sometimes people say, well, what about phenotype? Can the clinical phenotype help us distinguish between CFTR-related disorder and CF? And in 2011, with the first consensus statement, CFTR-related disorders were described as mono-organ. So it was, you had absence of the vas deferens, or you had pancreatitis. And if multiple organs were involved, it was felt that this was actually representing CF. But the problem with that is how many organs are involved depends on when you assess. So are you going to be assessing a 7-year-old with pancreatitis, a patient? Are you going to be assessing for absent vas deferens in a 7-year-old? Might not be so feasible. Also, your 7-year-old with pancreatitis may not have evidence of lung disease at age 7, but might have evidence of lung disease at age 27. So when you assess is important, and how hard you look is important, how many tests you're doing, and how you define involvement. So if you do a CT scan on a patient and you see bronchial wall thickening, how much bronchial wall thickening counts as evidence of a problem? And so the definitions of 1 organ, 2 organ, 3 organs is not so clear. So in 2022, the updated European guidelines for CFTR-related disorders state that involvement of more than one organ does not preclude a diagnosis of CFTR-related disorder. So that makes it very clear that phenotype is not going to help distinguish between CFTR-related disorder and CF. And there is considerable overlap with certainly patients that we follow with CFTR-related disorder having far more severe clinical phenotypes with regards to severity of lung disease, even at a relatively young age. So that severity of disease as well as number of organs doesn't help you distinguish CF from CFTR-related disorder. And the difference between these is not in the phenotype, but in the degree of CFTR dysfunction. Another knowledge gap or question that comes up is CFTR-related disorder the same as CF-SPID or CFTR-related metabolic syndrome? And these two conditions are ones, they mean the same thing, they're just used in different countries. And the short answer is no, they are not the same as CFTR-related disorder. CF-SPID refers to the situation where you have newborns screened positive but not meeting the diagnostic criteria for CF. And CFTR-related disorder is a clinical presentation of disease in a CF-related organ, whereas CF-SPID is a biochemical presentation in an asymptomatic infant. So CFTR-related disorder is disease, there's a problem. CF-SPID is asymptomatic, but there is evidence biochemically that something's off. But both these conditions may have a borderline sweat test and have CFTR variants. So there's definitely an overlap there. And the question is that we still don't have certainty on the prognostic significance of CF-SPID. We do know that some people with CF-SPID will go on to meet diagnostic criteria for CFTR-related disorder or CF, but the risk is still yet undefined, and we can't predict with certainty which children will progress and which children will develop a clinical phenotype or have a change in their measures of CFTR function with a sweat test that goes above the cutoff of CF. And often patients that were diagnosed with CF-SPID a number of years ago as CFTR2 identified variants of varying clinical consequence that were then redefined as disease-causing that caused changes in categorization. So the key thing here is that there is the potential relationship between the two, but they're not the same thing, and that infants with CF-SPID should be monitored for the development of CFTR-related disorder or CF. The next question that comes up is there another condition on the CFTR spectrum between CFTR-related disease and obligate heterozygotes? And it's sort of been a long-stated belief that carriers of CF are asymptomatic. But there was a recent publication by Miller that looked at 20,000 CF carriers and a control population and identified 57 CF-related clinical conditions that were more frequent in CF carriers. Now, part of the problem is that this did not consider the possibility that some CF carriers may have in fact had a second CFTR gene variant and may in fact have had CFTR-related disorder. And in a study by Esposito from Italy, they looked at 353 carriers, so the parents or siblings of people with CF, who then had full sequencing and assessment of clinical symptoms in those where they actually found a second variant. So familial variant, but then a second variant. And they found that 6.8% of these 353 carriers had a second variant and also had clinical symptoms. So there is this sort of not yet clarified sort of concept that there may in fact be something in between CFTR-related disease and obligate heterozygotes that in the European consensus report on CFTR-related disorder, they call CFTR-associated disease. And they demonstrate that with this graph looking at in vitro function along this sort of x-axis and the clinical genetics and biomarker results on the y-axis. And yet we at this point in time, this concept is just a concept without diagnostic criteria and still without certainty that this indeed exists as an entity. My last sort of question is, will CFTR modulator therapy help people with CFTR-related disorder? And in the guidelines that were published just last year, looking at variant-associated therapies, the quote is, for patients with a diagnosis of CFTR-related disorder, there is no evidence to support the use of CFTR modulator therapy outside of clinical trials. But we do know that CFTR function can be corrected with modulator therapy, and it does help some phenotypes of CF and also of CFTR-related disease, such as recurrent pancreatitis. And when we look at this nice summary table of studies done in people with CF who were either pancreatic sufficient or insufficient, we see that CFTR modulator therapy results in improvements clinically. And in those people who are pancreatic sufficient, CFTR modulator therapy can decrease the likelihood of and the frequency of acute pancreatitis. And depending on when CFTR modulator therapy is started, we can see that there is a rescue or recovery of some CFTR function in those who are pancreatic insufficient. So there's certainly evidence that recurrent acute pancreatitis is a CFTR-related clinical phenotype. So there is the theoretical possibility that modulator therapy may help people with CFTR-related disorder. And in fact, the fourth paper of the series of 4 papers from the European Consensus Conference actually addresses, as part of this, the question of clinical trials in people with CFTR-related disorder, sort of saying that some of the clinical syndromes would be similar to, you could use the same outcome measures as in CF trials, for example, people with bronchiectasis, where you could look at FEV1 and frequency of exacerbations, or people with pancreatitis. But some endpoints are harder to assess because we don't have as good a knowledge about the rate of progression of CFTR-related disorders and some of the potential risks that we might see with this condition, like the potential for increased risk for GI malignancies. It's very difficult to assess that. So I just wanted to end with one sort of example and kind of real life example of a patient that was referred to us because of CBAVD. But when we took a history, we realized that there was evidence of lung disease and sinus disease and also a family history that suggested CF. And testing also indicated that there was significant lung disease and presence of bronchiectasis. But the genetics, even after full sequencing, only showed one variant. So we couldn't diagnose CF on the basis of genetics. And not surprisingly, given the variant that was found, the sweat tests were borderline. So we couldn't make a diagnosis with the information we had. And so this could have been CFTR-related disorder, or it could have been CF. Fortunately, in Toronto, we have access to nasal PD, and so testing revealed that on a second functional test, there was no response to the chloride-free and isoproterenol. So this was looking like a CF pattern, and so we were able to put a label of CF on this patient, and he followed this diagnostic algorithm. However, regardless of the diagnosis of CF or CFTR-related disorder, we all know how to treat bronchiectasis and Pseudomonas, et cetera. And after applying standard CF therapies, we could see a clinical improvement. This was 14 years ago, and patient is still doing well. So in conclusion, CF and CFTR-related disorders are along a spectrum related to dysfunction in CFTR, and CFTR-related disorders are still kind of unrecognized or misclassified. And we still have challenges, very practical challenges in making the diagnosis because of the testing requirements. And we still don't fully understand the natural history of CFTR-related disorder in terms of progression over time. And we also don't completely understand the link between CF-SPID and CFTR-related disorder. There is a role, however, for CFTR-related disorders registries, and so that we are capturing information on this patient population. And in Canada, this is part of the CF registry with a separate tab that collects all the same information as the CF registry, but identifies someone right from the word go as a diagnosis of CF or CFTR-related disorder. And then the final one is that this is an important condition to consider, especially now as we go into looking at treatments that can help correct CFTR dysfunction. Thank you very much.
Thank you, Dr. Tallis. We do have 2 minutes for questions. If anybody has a question for Dr. Tallis, please come to the microphone in the middle here.
Jennifer Logan, Lehigh Valley. Can you comment on the propensity for some kids or people in the gray zone, the 30 to 60, to change their sweat chlorides over time? I find this the most frustrating thing.
That's right. Well, I mean, that's a great question. I mean, their sweat test is can change. There's a variability associated with the test itself, and the degree of variability depends on if you're in the high range, the less function range, or the lower range. With a bigger variability from test to test, the more closer to normal you are. But diet and environment can change your sweat test results, and time, aging also tends to result in the sweat tests getting, values getting higher. So it is
both going up and down with age.
Yes, and so the variability can be plus or minus 15 points, you know, within from test to test. And so you could, if you're in the borderline range and you go minus 15, now be in the normal range, or plus 15 and potentially be in the CF range. So that's part of the reason why for CFTR-related disorders that they recognize that the sweat test alone is not adequate enough to measure CFTR dysfunction.
Thank you.
So I have a question. Still 1 minute. I wanted to ask about the last patient you described, and you said that gene testing had been done. How extensive was that?
It was full sequencing.
Full sequencing.
Yeah.
And,
Yes.
and for rearrangements and
Yes.
everything. Yeah.
And has been repeated because, you know, every few years we're like, oh, we should do this again because we haven't found a variant, second variant. Yeah.
Hi, my name's Georgiana. I'm a genetic counselor from Iowa. And I was just wondering, when we think about, like, you know, newborn screening with some sort of genetic screening and then a sweat testing clinic, is the sweat test still the gold standard for diagnosis of cystic fibrosis? Or given that there are, you know, CF-related disorders in this spectrum, should we be thinking about different types of testing as part of our process?
That's a great question. I think some of these questions regarding genetic counseling, et cetera, will come in with the next speaker. But the sweat test for the diagnosis of CF is still a very important test in conjunction with genotyping. So we're not looking to change it as a diagnostic test for CF. As a test for CFTR-related disorder, it is problematic.
Hi, my name is Kaya. I'm a research coordinator. You mentioned the lack of resources for testing with MPD in North America as well as Europe. What do those numbers look like in Asia, the Middle East, and Africa? And what steps are being taken to increase availability of that testing?
Well, the problem with the nasal PD and intestinal current measurements is they're very challenging tests to do. Nasal PD requires a trained technician, a nurse, or coordinator to do that. And there's lots of reasons why you can get problems with tracing. So nasal inflammation, if the person's not staying still, smoking, there's all sorts of things that can affect it. And so it's a very technically difficult test to do. So in countries where CF is not so common, a lot of places in the world don't have access to sweat testing. And so that really is kind of the first bar to reach. Then the question comes up is, should nasal PD actually be available more broadly, or should it be recognized to be complicated enough that it should be in sort of specialized centers where patients can be referred to the center to have it done, because the kind of quality control that's necessary to get a good test strip is difficult, is challenging.
Thank you.
Thank you. Okay, so we'll have our last speaker today, who is Eleanor Langfelder-Schwind from New York, genetic counselor at the Cystic Fibrosis Center there. And I will hand it over to her.
Advancing the slides. All right. Hi, everybody. Thanks for staying with us. I am just so grateful to have the opportunity to speak as part of this fantastic panel. I have no conflicts of interest to disclose, but some of the work I'll be discussing was funded by the Cystic Fibrosis Foundation and a Vertex Innovation Award. I also have to disclose that being invited by Joanna Ramans to speak at an NACFC panel on the 35th anniversary of CFTR is really a thrill. So thank you so much to you and to Samantha for inviting me to be part of it. Given the recent proposal to add genetic counselors as core team members and on CF care centers, I thought I'd start with this quote. The necessity for frequent interaction between a CF family and a genetic counselor is best accomplished when the counselor is a member of a team of health professionals working together in a CF center. But you won't find this quote in the recently released position paper. It's actually 50 years old, and it was written by some of the pioneers of CF care and CF genetic counseling. Dr. Carolyn Denning, who founded my center and several other centers in New York, was actually trained by Dorothy Anderson. So I like to think about the provenance of this quote, that it goes all the way back to her. So genetic counseling is the process of helping people understand the implications of genetic contributions to disease. And typically it involves interpretation of family and medical histories, genetic education, and counseling to promote informed choices and adaptation to the risk or condition. Genetic counselors typically have advanced training in medical genetics and counseling, a master's degree from one of now 60 training programs in the United States and 5 programs in Canada. And in the US, we're certified by the American Board of Genetic Counseling with over 7,700 certified genetic counselors and licensure in 34 out of 50 states. Our professional organization, the NSGC, has a peer-reviewed journal, a code of ethics, and professional practice resources, including a Cystic Fibrosis Special Interest Group, or SIG, which now has over 100 members. We meet quarterly, and we met this morning here at the NACFC. But unlike our colleagues who are social workers and dietitians and respiratory therapists, genetic counselors are not recognized as independent providers by the US Centers for Medicare and Medicaid Services, or CMS. And apologies in advance to the Canadians and the Europeans in the group. I'm going to talk about this for a couple of minutes. Recognition by CMS is important for billing, and especially for coverage by Medicare, and then it sets precedents for other insurance providers. And this is important not only for the aging percentage of CF patients who will need our services and will have Medicare, but also a lot of the patients that Dr. Tullis described who have CF, are being evaluated for CFTR-related disorders, who have bronchiectasis and are coming to us as an older age. I'm grateful to the CFF who signed on as a supporter of the bill, I believe 2001 or 2002, but that bill still needs additional supporters. So yes, I see people scanning the QR code. If you are involved in a society or an organization that might be a supporter, please reach out to me or really any genetic counselor that you know. They will be more than happy to help you. Genetic counselors work in more than 20 different specialty areas and typically can provide services in person or by telehealth. Some common misconceptions about genetic counselors: we do not require or promote genetic testing. We do discuss testing options and possible results to ensure informed consent, and also explain that genetic test results may not always provide a clear answer, as we saw from the last speaker as well. We are not licensed therapists, but we do use our counseling skills to establish rapport and provide support during the genetic counseling process. And we do not make recommendations about whether a person should have children. We do provide and interpret information to facilitate family building decisions. We provide insights and anticipatory guidance to promote reproductive choice in keeping with the patient's values. So we elicit the patient's values and we have those conversations within the framework of those values. But sometimes these discussions can lead to perceptions of a values mismatch between the counselor and the client. And this is a priority area of research for the National Society of Genetic Counselors to understand how we're communicating about reproductive choice. So turning to what's happening in the CF care centers, about less than a quarter of CF care centers actually have named a genetic counselor that's associated with their care center in their 2023 CF Care Center report. So that's 65 out of 287 programs across pediatrics, adults, and affiliate centers, and that's actually 44 unique names, so some genetic counselors are clearly covering more than one program. The CF Foundation's patient registry asks one question about genetic counseling, and that is whether or not parents of children under the age of 2 had genetic counseling. And this is by any provider. It's not specific to whether or not they met with a genetic counselor. And for babies born between 2018 and 2020, 42 to 47% had genetic counseling as per the CF Foundation registry. And as Samantha mentioned, I'm from New York, where we have a CF newborn screening algorithm that includes CFTR sequencing and often complex discussions and family testing to determine phase. And in our cohort of infants with CRMS CF SPID, which is about 225, we might be down to 223, about a little over a third of families met with a genetic counselor. So with support from a screening improvement project from CFF and some encouragement from Dr. Phil Farrell, who's been a really wonderful champion of genetic counselors for a long time, a group of us looked at perceptions of the knowledge and value that genetic counselors bring to CF care centers. And so we looked at whether or not these centers were engaged with a genetic counselor, closely working, whether there was a referral relationship or there was no contact between the CF care center and genetic counselors at all. And maybe not surprisingly, but what we were able to show is that centers that have engagement with a genetic counselor were more likely to agree that genetic counselors provide unique and valuable services and should be part of the core CF team. Genetic counseling needs are there at every life cycle stage, not just in the prenatal and in neonatal, but I will go through this. It's not meant to be exhaustive, and if other genetic counselors want to come up and share some of the things that they do with families, that would be great. But a typical referral indication is with carriers of CFTR variants who are maybe at a 1 in 4 risk, or who have a positive prenatal diagnosis. And in those scenarios, we facilitate informed reproductive decision-making, based on current data about genotype, phenotype, and penetrance and natural history of CF and treatment options. But a study by Elsas et al. From 2017 looking at how prenatal genetic counselors might speak with their patients who are maybe expecting a baby with CF, they were not comfortable talking about treatments themselves, but were referring to the care center. And now with CFTR modulators available as early as 1 month and a growing interest in prenatal treatment with CFTR modulators, it's really critical that genetic counselors have a very good understanding of what treatments are available and feel comfortable talking about them and then referring to the CF care center for further discussion. In terms of the neonatal period, the benefits of genetic counseling in CF newborn screening are very well established with lots of publications from all over the country. But new recommendations for adding a CFTR sequencing tier across the country will increase the need for genetic counseling and CF expertise. And that because when you introduce the CFTR sequencing tier, we add a greater likelihood of some diagnostic dilemmas, the CRMS, CF SPID, due to identification of variants of varying clinical consequence and potentially variants of uncertain significance. Although not from CFTR2, but as defined by the ACMG criteria that might be used if the variant is not yet in CFTR2. In childhood, and a lot of these, the things I'm going to talk about kind of carry through to adolescence and adulthood, but periodic review of CFTR genotypes, making sure that patients are well documented in their genotypes, that we know what research studies they might be eligible for. Genetic counselors are very well suited to assess whether rare CFTR variants may be responsive to modulator therapy and assist in making those arguments for that off-label use. We have age-appropriate discussions of CF genetics and genotype and discuss future family building with parents. We can offer genetic counseling outside of the immediate family to relatives, and then also, which can be quite helpful, is coordinating testing for CFTR-related disorder evaluation and the differential diagnoses, which can include primary ciliary dyskinesia, which is another genetically complex condition that requires a lot of interpretation and sometimes patience. And then all of this in the adolescence, we can begin conversations about fertility and family building for the adolescent with cystic fibrosis as part of the team. And then into adulthood, we would continue those conversations in addition to everything above. And we know, and we're about to go, I think, to a plenary session about reproductive and women's health. And we know that reproductive care is an essential need in the CF community. People with CF can envision a future where they have families of their own and they want to learn more about the scope of family building options. So genetic counselors can, among other things, discuss CF-specific screening and testing options, including standard recommendations outside of cystic fibrosis, as well as expanded screening. And then for the people with CF, we can review their medications, pregnancy and breastfeeding data, and coordinate with CF newborn screening programs and pediatricians for the follow-up of infants and born to women taking CFTR modulators during pregnancy, who are known to have potentially a false negative newborn screen. So with all of these genetic counseling needs and really not a lot of access, what can we do to improve it? Well, there actually have been a lot of policy statements before this recent position paper, but they all kind of they're more about mentioning genetic counseling, whereas the new position paper really is very specific about genetic counselors. So until this new paper came out with a much nicer and more complex diagram, which Charlotte Close went through really beautifully yesterday in the big session, genetic counselors had typically been kind of on the outer circle of CF care providers, and based on these new recommendations, they moved into the inner circle, where they have a much closer connection both with the CF care team and with the patients and families. And according to these recommendations, the genetic counseling by the genetic counselor can occur either at the home institution or in creative ways of making connections, such as via telehealth. And so that brings me to talk about another screening improvement projects funded by the Cystic Fibrosis Foundation that I was not involved with, but it's a wonderful project, and I think some of the people are here. to provide genetic counseling via telehealth to families with a positive CF newborn screening result, where the counselors are sitting in Florida and the patients are having a sweat test in Connecticut. And if you haven't seen the paper, you should definitely take a look at it. And these had very high levels of patient satisfaction, as well as an increase in adherence to sweat testing recommendations when that genetic counseling was offered in conjunction with the sweat test on the same day, among many other things. So that, in combination with the pivot to telehealth during the COVID-19 pandemic, led to some more opportunities to really explore the feasibility of centralized CF-specific genetic counseling services via telehealth outside of the newborn screening realm and into the CF care centers. So we began a 2-year project in 2022 that we're just finishing up on, although of course the planning took a lot longer than that, but this would develop a telehealth-based delivery model for providing CF-specific genetic counseling and assess the feasibility. And we're calling it CFGCT. It's not a great acronym. If anybody has a better one, we are really open to suggestions, let me know. So we want to determine the essential elements of the CFGCT, develop the model referral system, and then send it back for broader feedback via survey. Put together a terrific team across 4 institutions and again, a wonderful advisory board, Mel Collins, who was part of the Florida-Connecticut connection, and we have insurers, genetic counselors, and also patients, as well as parents of people with CF in our group. And it was a mixed method study. We held a series of focus groups of key community members. We leveraged a fantastic implementation science team. And boy, have I learned a lot over the last 2 years. Designed the model with input from a telegenetics consultation service so that we could get a handle on the financial and the logistical needs. And that's more about that can be found poster 382. We held 15 focus groups and with a variety, again, of key community members, clinicians, program coordinators, genetic counselors, newborn screening staff, parents of people with CF, as well as parents of children who've had a positive newborn screen, partners of people with CF. That was a new fascinating group to learn more from. And then we were able to have a focus group of insurers as well. And just a very brief summary of our key findings and themes. The clinicians and the coordinators who might be referring patients to this type of service, they really focused in on whether the genetic counselor was a knowledgeable CF-specific genetic counselor. And the patients wanted access to accurate and digestible variant-specific information that, again, that was tailored to their needs and values. And I would say the parents in particular were very firm that this had to be equitably accessible, that cost, and that it needed to be low cost or no cost for people who were not able to afford it, and it needed to be accessible. And some of the key facilitators were connection between the program and their home CF care center. They didn't want to go outside their center, and connections with the Cystic Fibrosis Foundation, and again, the perceptions that the genetic counselor was knowledgeable. So we prepared a video summary of the CFGCT model and sent it out with help from the CFF to care center physicians and program coordinators, as well as the community member focus group participants or people who had expressed interest in being in the focus group but were not able to attend. The QR code is there. Again, it's also on my poster. The survey is closed, but if you listen to it, the narration is familiar because Karen Rary did the voiceover. She's very good at it. And our feedback survey, we heard back from 67 care centers. It's about 23% of the 287 programs. We had nice representation across adult, pediatric, and combined centers, and also a good representation across whether or not the centers had access to a genetic counselor. So some had a genetic counselor on the team, some referred to somebody they were comfortable with, some had generalist genetic counselors that they didn't refer to very often, and a few centers had no genetic counseling access or genetic counselor access at all. So we asked them, we asked them a number of things, but we asked, would you refer your patients to this program? And about 70% agreed. So of course we asked why not of the others, and we found that basically the 100% of the people who said they would not agree already have a CF-specific genetic counselor that they were working closely with and were really happy with the services that they were providing to their patients. Some wanted to learn more about program implementation. Some had questions about billing and reimbursement. And then 2 center staff responded that they were comfortable counseling families themselves. I want to just take a quick pause there because we expect that CF clinicians are comfortable talking about genetics with their patients. It's a common inherited condition. We expect it. In the same way, CF clinicians are also comfortable talking about nutrition, and they're comfortable talking about airway clearance, and they assess and address the emotional needs of their patients. And the rest of the team assists and provides maybe extra depth and more conversations. And I think genetic counselors can kind of fit in the same mold. It doesn't mean that clinicians shouldn't be talking about genetics. They should, because we want to make sure that there's consistent messaging, and patients sometimes need to hear about the genetics and anything really in a few different ways at a few different times. Very quickly, the community results, we had 46% of the respondents return the survey. Again, a variety of people with CF, parents and spouses, et cetera. And we asked them, would you like to use the service? And 73% agreed. And again, the ones who disagreed tended to already have access to a knowledgeable genetic counselor. Some had questions about billing and reimbursement, and some were not planning to have more children, which means we need to do a little bit more work in helping people understand other reasons why one might want genetic counseling besides in the reproductive space. But when we asked, would you refer friends and family to the service, the answer was overwhelmingly yes. And one of the quotes that came back from the community was, I am a mother with CF, so I know my kids are carriers, but I would like this program to be available to my kids and their partner down the line for reproductive decisions. And I have to say, like, that just touched my heart a little bit because, you know, we're in a world where we're talking to women with CF about having children, and now they're thinking about their grandchildren too. And that just, what a great time to be in this field. So with community feedback and developing a model, our next step is hopefully to implement a pilot study. So we will report back sometime in the future. So just in my last 10 seconds, just a couple of takeaways. So patients and families want genetic counseling comprised of that accurate, variant-specific information that's tailored to their needs and values and delivered equitably. And as the spectrum of CF and CFTR-related disorders continues to expand and the phenotypes are more varied and uncertain, as we've heard today, the need for genetic expertise in CF care centers is just going to keep increasing. Genetic counseling for people with CF and CFTR-related disorders is enhanced by connection and engagement between genetic counselors and CF care teams. And I'll put it out there that the skills of a genetic counselor with CF training and expertise are complementary to the other CF care team members and viewed as value added when the genetic counselor is a member of the CF care team. And for centers without an on-site CF genetic counselor, access to CF experienced genetic counselors may be improved with a centralized CF-specific telegenetic counseling service, which may or may not be called CFGCT. I thank my really wonderful team who lets me take care of their patients too, as well as do my own research. The wonderful CFGCT team and the CFF and our fantastic advisory board. And I'll take questions.
So we do have a couple of minutes for questions for Eleanor before we will turn it over to the whole panel.
Hi, Eleanor.
Hi.
I'm Mel Collins. I'm from Connecticut, University of Connecticut. My question for you is, with the new modifications proposed to the newborn screening guideline, how would you envision genetic counseling fitting into that? For example, our algorithm, we generate a lot of genetic counseling in our program because we're doing it at the time of sweat for a baby with a single mutation. And I feel like there's always two Mels. There's the newborn screening Mel who finds CF SPID to be the bane of her existence, but then there's CF Doc Mel who sees families who didn't know that they could have a child with CF because their other child only had one mutation and screened negative. So I'm just trying, I know that's like a philosophical thing.
All right, well, I will do my best there. So as you know, every state is going to do this, implement this a little bit differently. And even within states, I mean, even in New York, where we've been doing this sequencing for a long time, centers are doing things differently. And we are just taking steps to make sure that there is a genetic counselor in every care center in New York. We have a QI project, a SIP-funded QI project to do that. When we in New York changed over from IRT DNA to the sequencing algorithm, our one mutation referrals were eliminated. And the way that it works in New York is that families and practitioners primary care practitioners are notified of the carrier status so that the families can seek genetic counseling. At the time, both because most centers didn't have a genetic counselor and also because we kind of came to the conclusion that the families didn't necessarily need a CF-specific expert genetic counselor. They needed a prenatal genetic counselor who's going to take care of their reproductive needs. We thought, unless they have questions, in which case come back to the CF center, of course. So I think it's going to be implemented differently in different places, but there needs to be a plan for it. So in terms of the CRMS CF SPID referrals, they are difficult genetic counseling sessions for sure, and it's not just one, it's definitely something over time, and the CRMS CF SPID guidelines do also create a space for genetic counselors to be meeting with the families over time. So I think they are fewer in number, but they are very anxious, and honestly, the reproductive counseling is fraught with uncertainty because we have a healthy baby in front of us, and you could say you never know what the future will hold, but in this case, we want to take all the best available information that we have and counsel the families that they can move forward in a way that works for them.
I'm going to invite all of the speakers to come back up. We do have a couple of questions online, and we also welcome anyone to ask more questions now. We have about 10 minutes.
Yep. Right here.
Hi, Brad Kwan, Vancouver, Canada. I have a question for the first two speakers. Are there any plans to integrate some of the in vitro response data that you're identifying into CFTR2, recognizing that there may be some challenges in interpretation by patients versus clinicians?
The short answer is yes, with the caveat that we don't know exactly how, when, and what that will look like. This has been discussed amongst our team for quite a while, and there have been some conversations about, there are lots of reports in the literature about in vitro response data. Which ones do we pick? And, do we need to vet things like that? So I think, and Joanna, you should weigh in as well here, on the CFTR2 team, I think we have talked about the idea of putting that type of response data online, and I think we'd like to, but we don't yet, I think, have a timeline for that.
Yeah, but also remember the real strength of CFTR2 is the patient data, and that has to be in sync. And it's not always in sync, but it would be, and we wouldn't want to do it without integrating that very carefully. And also now population data, although sometimes that isn't definitive, but it's helpful.
Yeah, I think one of the things that we also need to think about is, there are lots of variants that respond to ETI, especially variants that have a lot of function and probably don't cause CF. And so, and some of them are on label and we know they don't cause CF. And so there's also, we just need to be very mindful of the type of information that goes online. So for the time being, for those in the room, we do have information online about whether a variant is on label in any country. And it'll just say, some patients may be eligible for X, Y, and Z. So that information is there. So far, we haven't ventured into things that are off label, but it's certainly an area of discussion for our team.
Great, thank you.
Hi, I'm Eitan Kerem from Jerusalem, Israel. I have a question to Dr. Biller. First, I want to congratulate for this very, very challenging and important data that enabled many, many patients to benefit from the ETI, and for them, it was life-saving. However, the FDA determined 10% threshold is very, very important in a community, in a population study. But when you come to the individual patient that had response in 9.95%, whether this patient is ineligible, and we need to define criteria for those patients when they can be eligible. For instance, in my country, if we show response in organoids or even clinical response after 1 month, they might be eligible to receive ETI refundable. And the second question is regarding the 5 mutations that, according to Vertex FRT studies, are responsive, and according to your studies are not responsive, if you have clinical data to know whether they are clinically responding or not.
Okay, to the second question, I don't know the clinical data for those. I know they're approved, which is, I guess, a good thing. And the first question, I think we have to take all the evidence into account that is out there to see if it would make sense to give someone a treatment. I'm not sure if this answers your question in full, but
it's just, I think, a general comment that we need as a community to develop some guidelines for patients that carry mutations that are ineligible, but maybe are close to the 10% threshold.
Yeah, as I said in my presentation, there's enough evidence that people benefit from a few percent improvement in their baseline function. Yeah? And if these benefits are, they are real, then in my opinion, they should be on treatment. Yeah, and sometimes I think you just have to give people, the drug to see if their symptoms improve, and I think that's way better evidence than discussing, if a 10% responsiveness cutoff or approval threshold makes sense. Yeah.
And I have a question to Liz regarding your interesting patient. In the paper showing exon 9 skipping at that time, exon 10 skipping, showed that patients that have homozygous 7T had up to 30% exon skipping, and even those with 9T had up to 15% of 9 skipping. You have maybe data on TG repeats or something like that, showing that this is actually the other allele that is causing CF in this patient?
So that's a great question. It's not in the reports that we get from genetics at Sick Kids, they don't report the TG repeats with the T-tract, and you have to go back and specifically request it. So I don't have that information at this time, but that's a great comment.
Brian O'Sullivan, Dartmouth. I actually have a follow-up for Etienne's question that Marty Solomon's paper in Lancet Respiratory Med, look at N1303K, showed that they did have a response over 10% with the FRT testing in vitro. So it seems like there's different results with different processes with these tests, and how do we unify that so that we're all looking at the same results? And if we have to use a cutoff of 10%, which test do we use? The one that shows 12% or the one that shows 9%?
It's a good concern, because there are also some variants that test a little different in a different assay.
Yeah, and I have to say, in our assay, N1303K only missed the 10% responsiveness threshold by 0.6%. So, and it's to be honest, a low-powered study with an N of 6. So it could have easily crossed that 10% threshold, and then, we would have similar results.
I'm sorry. I'm a clinician. I don't know all this stuff all that well. So, when I see Marty's group reporting one thing and other groups reporting something else, I just get confused. And I didn't know if there's a process that, like NPD has had to be unified across centers. I don't know if this FRT testing is different in different labs or needs to be unified somehow. I'm asking a question because I just don't know this area.
And I mean, FRTs, that's a mutation-specific approach. If you use H&Es, you're looking at patient-derived cells. Yes, I'm sure there's more standardization that is needed, and also defining meaningful thresholds is certainly something that is, much needed, I think.
Okay, so that's our time. But we'd like to please join me in thanking all of our speakers today.